can start.
Welcome to biology practical class. This class is a preparation class for WAEC 2024 biology practical examination. We have been given a mandate to prepare 16 specimens. So I'm going to take you through the 16 specimens. Thereafter, we take them one after the other. So these are the specimens that we are told to prepare. We have specimen A, which is gill of a fish. Specimen B, whole liver from a domestic fowl. Specimen C, lungs from a domestic fowl. Specimen D, whole gizzard from a domestic fowl. Specimen E, whole liver from a domestic fowl. Specimen F, ginger rhizome. Specimen G, onion bulb. Specimen H, leaf of biophyllum. Specimen J, cassava stem. Specimen K, cassava tuber. Specimen L, thoracic vertebra. Specimen M, humerus bone. Specimen N, mango seed. Specimen P, femur bone. Specimen Q, lumbar vertebra. Specimen R, orange seed. Altogether, it makes 16 specimens. So we are going to be talking about the first one, which is what? Which is specimen A, gill of a lizard. If you look at this diagram, is a drawing of gill of a fish. Gill of a what? Of a fish. So there are some things that we are expected to know about this specimen. The very first thing we are going to be talking about on this specimen is the location of the specimen in a fish. The location of the specimen in a fish. Location. Where is it located in a fish? It is located below the operculum. Below the operculum. Or say gill slit. Gill slit. The second thing we are considering is the habitat of the specimen. What's the habitat of the specimen? The habitat is aquatic habitat. Aquatic habitat. Aquatic habitat. But don't just write aquatic habitat. You have to give us examples of what? Aquatic habitat. So you can write river. You can write stream. etc. Like so. The next thing, let's talk about the phylum of the specimen. Phylum of the specimen. The phylum is chordata. Chordata. That means they have a backbone. The class of the specimen now. The class is class Pisces. Pisces or fishes. Is that you call it? Pisces or what? Or fishes. Let's also talk about the biological significance. Biological significance of the specimen. Of the specimen to the organism. For respiration, that's the biological significance or function. Respiration or say exchange of gases or breathing. You can be asked those characteristic features that adapt this specimen to its function. Characteristic features that adapt this specimen to what? To its function. So this is a respiratory organ. The gill is a respiratory organ. So it has some characteristic features that adapt it to its function. So let's talk about those characteristic features. Characteristics. adapt the specimen to its functions. Number one is moist or wet surface. Moist or wet surface for dissolving of gases, e.g. CO2, oxygen. That's the first one. The second one is thin surface membrane for easy dilution. What is the dilution of gases? The third one is highly vascularized. That means it is supplied with a lot of blood vessels. Highly vascularized for transportation of dissolved gases or food. That's about the characteristic features that adapt the organism or the specimen to its function. Lastly, let's talk about the observable features of this specimen. What are the things when you see a gill? What are the things that you can see in a gill? What are the observable features? The first observable features. Number one, we have red in color. It's red in color. Number two, we have presence of gill filament for exchange of gases. The next one that you have is gill recars to prevent entry of food particles. You also have gill arc that provide support for gill filament. Okay, you can be asked example of organisms that belong to class Pisces or fishes. Example of organisms, such organisms, you have tilapia, you have shark. So these organisms belong to what class? Pisces. You can also be asked that you should give examples of organisms that use gill as the respiratory organ. Crustaceans such as prawn, crayfish, crab, they use what? They use gill for their what? Respiration. Fowl, liver from the domestic fowl. Location, where is the location of this specimen liver in a fowl location. The location is lower part of the abdominal cavity. Lower part of the abdominal cavity. Let's talk about the habitat of the specimen. Habitat, that is terrestrial. Terrestrial habitat. Like I said earlier, try as much as possible to give example of terrestrial habitat. Say land, garden. Garden. Let's talk about the phylum. Phylum, that is for data. Class, hips, or birds. Hips or birds. Observable features of the specimen. One, what are things that you can see from the specimen? Number one is red in color. Two, it is soft to touch. Another thing is that it has oval shape. It has oval or oblong shape. Now, we are told that the liver that we should prepare should have attachment of gallbladder. So you can include it. Gallbladder is attached. Gallbladder is attached. You can ask it, but now let's talk about biological significance. Biological significance of the specimen. Biological significance of the specimen. Biological significance of the specimen. Number one. What is the biological significance of liver to a fowl? Number one, it stores glycogen. Number two, manufactured by. Number three, excretion of nitrogenous waste. Number four function of specimen or biological significance of the specimen. It production of heat for constant body temperature. And it stores vitamins and mineral salts Store vitamins and mineral salt. These are the biological significance of the specimen. Now, I said earlier that they asked us to prepare a specimen, which is liver, that has an attachment of gallbladder. I checked through the past question. I saw where the similar specimen were asked to prepare. Now, in that past question, students were asked to pierce the gallbladder and tear the substances that is oozing out from the gallbladder. Now, from the gallbladder, when you pierce it, the substance that will ooze out is what we call bile. 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 The substance that will ooze out is called bile. Bile. When you pierce the gallbladder, oozing substances now. The substance that comes out, bile, ooze out. Bile oozes out. When you pierce gallbladder, bile oozes out. And that bile, you might be asked, because students were said to be given red litmus paper and blue litmus paper. So you might be asked to test the bile. You might be asked to test, you might be asked to use the litmus papers to test the concentration of the bile. Now, bile is alkaline. Alkaline, that means when you use red litmus paper, it will turn red litmus paper to blue. And when you use blue litmus paper, it will be unaffected. It will be what? It will be unaffected. Now, you can also be asked the functions of bile. What are the functions of bile? Functions of bile. Functions of bile. Functions of bile. Number one, for emulsification of fats. Number two, add water to food. Number three, prevent decaying of food in the intestine. Number next, now neutralize the acidity of food. That is the acidity of food. So these are the functions of what? Of bile. So the functions of bile. Next one, please. Specimen C. Specimen C is lungs from a domestic fowl. Lungs from a domestic fowl. Now, you will recall that we talked about the habitat, the phylum, the class of liver, that means specimen B. So ditto is for specimen C. All what we said under specimen B for habitat, for phylum, for class is the same thing for specimen C. Because specimen C specimen B, specimen C, specimen D, specimen E. They are all got from what? They are all got from a fowl. So therefore, they have the same phylum, they have the same class, and the same habitat. They have the same phylum, the same class, the same habitat. So we are going to move to observable features of lungs from domestic fowl. Observable features. What are the things? Observable features. The first value tells that red in color. Another thing is soft to touch. It's soft to touch. It is moist or wet surface. Moist or wet surface. Let's talk about the function or the biological significance of lungs to a domestic fowl. Biological significance of lungs. Of lungs is for respiration, breathing, or exchange of gases. So you can be asked the characteristic features that adapt this specimen to its function. The characteristic features that adapt this specimen to its function is the same characteristic features that adapt gills to its function. This is a respiratory organ. Gill as well is a what? Is a respiratory organ. So check under specimen A to see the features that adapt this specimen to its what? To its function. Let's move to specimen D. We have gizzard from a domestic fowl. Observable features. What are the observable features? What are things that observable features? Red in color as well. Red in color. This one is hard to touch, and moist or wet surface. Now, what is the biological significance? Grinding food for grinding